this is chlorpridine, and you can see the homogeneity to red, right? Right. Then skip. CH3 becomes CHO, right? Group A to B, or binary group becomes CHO, right? Group AD. Okay? It's a big shift, right? In the adjustment property. Big changes. Why this? Huh? No. Not oxygen content. If the oxygen is had electronegative, no? What happens? It pulls the electron. Okay. Alright. So it pulls the electron from the pi ring of the apparatus. What the tetrapig of pi electrons, right? And what it pulls it, so there are certain changes in electron structure of the molecule. So that changes the absorption spectrum. As I told you last time, if you know the electron structure of the molecule, you can, and in the Bohm quantum mechanics, you can calculate the absorption spectrum. Okay? So you don't have a visual. Okay? So it's all reflection of how what is electron structure. Absorption also fluorescence. Fluorescence also the same. Okay? Fluorescence spectrum is also determined by the atom structure of the molecule. The quantum field of this fluorescence is also determined by the electron structure of the molecule. And that may change in the environment. Quantum field of chlorophyll A in 80% acetone is not the same as the quantum field of chlorophyll A in a leaf. Okay? Much lower than in chlorophyll A in transport. Okay? Because absorption fluorescence property decreases. If you see tryptophan or in a protein, tryptophan or protein, okay, its absorption properties, quantum field also changes. Quantum field is quite high for tryptophan in around 0.3, tyrosine around 0.2 or 0.28 or 0.3. But if you put the same tyrosine in a protein, it drops 0.3, 0.03 or 0.05. Around 90% drop in the quantum field. Okay? So all is so most of the proteins will absorb in 280. Fluoresce at where? 350. Okay. Why does that 350? Tryptophan, right? Tyrosine, why does it absorb? Which are they? Around 270. And that fluorescence around 310 nanometer. Up to 30 nanometer. Okay. Anyway, so that is because the fluorescence is mostly at 350. Although they are tyrosine, the quantum of tyrosine drop very severely when it bound to it. Okay. You can see tyrosine fluorescence if you can either optically silence tryptophan by modifying tryptophan, by tetranitromethane or something, they modify tyrosine or tryptophan, you can see that. Maybe NATA will modify tryptophan. Okay, optical silencing. Or there are many other means also you can see. Can you see tell me a protein which does lack tryptophan? Proteins that lack tryptophan, no tryptophan. lactoside doesn't contain any tryptophan, only tyrosine. Okay, calmogulin, okay, insulin, okay, they are the proteins which do not contain any tryptophan. So they will fluoresce around 310 nanometers or 312, 13 nanometers. Okay, and they are observed also very low, 270, 267 up to 80, 60, 80 nanometers. Okay, okay. Let's do something else here. Okay, go ahead. Okay. Okay. Now transition from unicellularity to multicellularity, right? Okay. Two. All right. Oh, sorry. Most of these students are here, sitting here. So I thought I teach all of them. Transition from unicellular to multicellular. What is this? Okay. So that unicellular life, okay, eukaryotic life, born 150 billion of years ago, okay, eukaryotic. Then, after some time, in process of evolution, of course, a chlamydomonas-like organism developed. And then you can see a loose association of chlamydomonas-like organisms to form a loose association. Okay. As a forward, as a transition between unicellular to multicellular. Okay. You can see here Pandorina, or Pandorina, Nidorina, Gonium, Idorina. There are the transition forms from chlamydomonas. These are loose association of what? Unicellular cells. Okay. To a so-called multicellular cell, valves, okay, which have cytoplasmic bridge between them. Okay, cytoplasmic bridge, right? The is cell connected by cytoplasmic bridge in valves, right? So if you see the valves, the cytoplasmic bridge is there between the cells. Not only that, it's a multicellular organism which is there. Because when it moves, a certain group of flagella move like this in front, and just like and a certain group of flagella behind, will go this way. Okay. So the one is how they go opposite direction. The front in one way and back in another way. So you prepare from one to another by movement, right? The coordination between the cells. Not only that, there is a differentiation. You can see the valves. Okay, these are reproductive cells. Reproductive cells inside the valves called vegetative cell. The clear differentiation. Vegetative cell and the reproductive cells. Okay. Specialization. Gonidia, 32 cell embryo. These are 32 cell embryo. Okay, gonidia. Asymmetrical cell division, large cells, go to reproductive cell, small somatic cell. Okay. So instead of equal division, they undergo asymmetry. The smaller one becomes vegetative cell, bigger one becomes the reproductive cell. Okay. Alright. So in small cells, reg A gene is on. So therefore somatic cells are replaced. Somatic cells replaced. Last cell, lag is on. So reproductive cell, somatic cells. Okay. So somatic cells are replaced. So it becomes a somatic genes. Somatic genes are somatic, you understand that, right? Vegetative. Okay. somatic genes are suppressed, so in reproductive phase. Here, the gonadal genes are repressed, so it becomes the vegetative phase. Okay. So you can see a spatial separation up to different kinds of cells. What vegetative cells in one place, reproductive cells come going into different places in the colony. Okay? But in Chlamydomonas, of course, you also reproductive cells and a reproductive phase and a vegetative phase. But what is happening here? A temporal spatial is temporal separation, time. Okay? The spatial separation is a temporal separation, Chlamydomonas. Right? What happens? Rega is on somatic cell or bioplastic is pressed. Lag on, cell lag is on, reproductive cells or cell pressed. Reproductive on the control cells, somatic cells. So at a certain phase of time, the chlamydomonas cells are what? Chlamydomonas cells are what? Vegetative. Sometimes they themselves become reproductive. Okay? So it is temporal separation of vegetative phase and the reproductive phase in chlamydomonas cells. But a multicellular organism like volvulus, okay, where there is clear connection between two cells, intercellular connection by cytoplasmic bridge, there is a separation of vegetative cells and gonadal cells, but they are all taking place at different places of the cell. So this is a colony now, right? Okay. Now, in the process of evolution, this colony became the place, compact. Okay? And you got a system where you have now the two cells are in contact. Okay? So you have plasmodesmata, connecting same cytoplasmic bridge exists in a plant, right? Two cells are connected by plasmodesmata. You know plasmodesmata, right? The connection bridge between two cells through the cell wall, right? Right? So the plasmodesmata connects two cells, right? So there is no movement of nutrients or signals or hormones. Everything goes by a plasmodesmata from cell to cell. Here it goes from cell to cell by cytoplasmic bridge in the hormones. Okay. So unicellular to multicellularity also led to adhesion of the adhesion between two cells, connected by cytoplasmic bridge called plasmodesmata in plants. And in animal cells, there is the same thing took place, but they are contact cells. Two cells in animals are connected by contact cells. We call them contact cells in animal cells. Okay. Alright. In our body, two cells are connected, so called contact cells, where there is also a bridge, okay, between the two cells. Okay. So these intermediate forms are colonial organisms. Okay, they are only made a colony. Okay, but they are not connected by intercellular intercytoplasmic bridges. These four examples of okay. Then that slowly developed into a colony-like multicellular organism like volvulus, and they are connected by intercellular organisms. You can see the volvulus. One here in many colonies of volvulus, right? In a single, okay? Many colonies. Okay. Now the question arises. So chlorophyll is green. Why chlorophyll is green? Why is the chlorophyll green? Huh? Huh? Because it is observing green light, right? All the colors they observe all the wavelengths of light other than green, right? You can see here. Here, right? None of the observing green, you see. I told you that, right? But it's green, 500% here. They are not observing anything, right? So it's a big stretch of wavelengths where they are not observing light. Okay. Don't you think ideally chlorophyll should have been black? Ideally. Plants should have been black. Other than green. Why? Because they should observe all wavelengths of light. Right? They are black. When they black, a black plant, a black spot, or black color, any black color, observe all the wavelengths of light. Right? So plants should be ideally black, right? To harness all the wavelengths of light and convert it into food. Right? They can carry energy. But they haven't made their green. Why do you think so? Why there such big deficiency? There is no direct answer to this. Okay, I will give you some explanation. Okay. I don't have the direct proof what I am saying. Okay. Alright, I am going to say now. Then because we are talking about evolution here. Okay. Evolution doesn't have a direct proof. Okay. Okay. You know anaerobic world, okay, here. We don't have any black chlorophyll yet. Okay. There may be some other pigments associated in the organism which look like black but is not really black. Okay. Some very, very old cyanobacteria organisms would look black, but they are not really black. They cannot absorb green light. Okay. Okay, I am just going back and come back to you later. Yeah. So this is anaerobic world, right? No oxygen, right? Unoxygenic world, right? Okay. And what prevailed? Archaea, right? As I told you, archaea prevailed here. Archaea is primitive organisms, right? Very, very primitive bacteria. They evolved here. And have you heard of a bacteria called Halobacterium halobium? Halobacterium halobium. Its old name, the old name was. Its now name is changed now, but it's still known as Halobacterium halobium. There are many bacteria like that. Millions of them. Okay, thousands of them. What did they do? They don't have chlorophylls. They don't have bacterial chlorophylls either. They have but they are autotrophic bacteria. Or they can synthesize their food of their own. Okay. What to do? They have rhodopsin, bacteriorhodopsin. You know, when you go to nature, we feel very good, right? Why? Huh? Huh? Fresh air? Okay. Any other reason? Nature is green for us. Nature is green. Okay. And we are very nice at it, because what is this? Our rhodopsin neurons focused on green light. Okay. Our ability to see the green light is maximum. Rhodopsin absorbs maximally in the green. Okay. And our ability to see the blue light or red light drops drastically. Okay. All right. Very, very low. Okay. So with the least energy, we can see green light. Okay. So we are so comfortable in green light or looking at anything green. Okay. All right. Okay. Similarly, Halobacterium halobium, as I told you, has a bacteriorhodopsin. We have inherited rhodopsin, our rhodopsin from bacteria, anaerobic world. Okay. Rhodopsin that prevailed in billions of years ago, around 3.5 billions of years ago. Okay. We have inherited bacteriorhodopsin and converted to our rhodopsin. Okay. Slight change, not much change. Okay. In bacteriorhodopsin and our rhodopsin and I. Same, almost same. Structure is almost similar. Okay. We have selected nature, you know. Okay. Because that is a pigment that was selected for animal eye. Okay. Okay. Invertebrate eye, vertebrate eyes. Okay. All right. Invertebrate pigment, vertebrate pigments, essential landing up in human eye. Okay. Evolutionary. Okay. All conserved. Okay. And if you see, in anaerobic world, archaea prevailed, right? So it's full of archaea. Okay. And photosynthetic bacteria, or even cyanobacteria, developed below archaea in the ocean, right? What? Okay. If you see the red one, okay, that is the absorption spectrum of bacteriorhodopsin. Bacteriorhodopsin. Okay. And this is bacteriorhodopsin, structure of bacteriorhodopsin. Okay. Helix. It also retinol. Okay. Vitamin A, right? Okay. So retinal linked to lysine. So this hydrophobic core of bilateral lipid membrane. These are spanning the membrane. Okay. This is structure. And so the N-terminal end, C-terminal end, in the hydrophobic core, we have this retinol. Therefore, it's same thing. Same vitamin A is also in our eye, retinol. Okay? Okay? That's the chromophore, right? You see the retinol. Retinol. This structure. That absorbs the pigment, green light. Okay? Therefore, this is green. That is the absorption spectrum of that retinol. Okay? All right? And this is in the membrane. Lot of retinol attached here. This is a structure of bacteriorhodopsin. Trimeric structure of bacteriorhodopsin. It is trimer. Okay? And you can see retinols here. These are all retinols here, right? Retinol, right? You see three retinols in the structure. Okay. So they are all responsible for absorbing green light. Okay. And there is no difference between structure of bacteriorhodopsin and human rhodopsin. Same trimeric structure. Okay, same exactly almost same. Okay. Slight difference in the protein structure. Retinol remains same. Okay. So you say evolutionary conservation, not from, it's from archaea to human beings. Right? Billions of years. But see the conservation of nature. Okay. Not evolved. Okay. New pigments. Okay, conserved. Okay. Okay. So now when I say the earth is full of what? Archaea, which contain what? Rhodopsin, bacteriorhodopsin, that observes a wavelength like this, right? As I said, it is also focused on green, 540 nanometers. Where the peak? 540 nanometers, right? And it is absorption decreasing to 700 here and decreasing to 400 there. Okay? All right. So that's retinol, right? That observing there. And when these cyanobacteria, kind-like organisms, or even bacteria, photosynthetic bacteria, they evolved below the archaea, they did not, what all the light coming to the falling on the ocean, okay, all right, were absorbed by what? Archaea. Okay. What archaea could not observe, okay, collected below, okay, where what? This photosynthetic bacteria evolved or cyanobacteria evolved. Okay. So when cyanobacteria or photosynthetic bacteria evolved, they did not see the green light. Because when they evolved, they were green deficient in green light. Okay, because all the green light is observed by archaea, by retinol. Okay. So they evolved a pigment called chlorophyll or bacterial chlorophyll, which does not absorb green light. Okay. And therefore the evolution of selection of the pigment, that ultimately resulted in synthesis of chlorophylls in the plants. Okay. And they did lot of permutations, combinations in the process of evolution, and finally came up with a pigment called bacterial chlorophyll. Okay. And this bacterial chlorophyll did a fantastic job for them, okay, to absorb light and convert to chemical energy, although anaerobically, right, from hydrogen sulfide, acetate, right, they convert chemical energy, photosynthetic bacteria. Okay. Then these photosynthetic bacteria, okay, through the course of evolution, simply changed their structure, shifted the wavelength from 780 to 700, then 663 to chlorophyll A, 760 to 660, save 700 for chlorophyll D. Actually, if you put chlorophyll F, 770, 700, 720 for chlorophyll F, to 700 for chlorophyll D, okay, to 663 for chlorophyll A, or blue shift, shifting to smaller wavelengths. Okay, but still they did not absorb green light, because cyanobacteria also evolved around a little later. Okay, after the photosynthetic bacteria evolved. Okay, we believe that photosynthetic bacteria is the progenitor of cyanobacteria. Okay, photosynthetic bacteria, right, which are bacterial chlorophyll A and B, are bacterial chlorophyll A is progenitor of cyanobacteria. Okay, cyanobacteria evolved from photosynthetic bacteria. Okay, all right. So they simply did some modification of side chains and shifted their wavelengths to smaller wavelengths, so they can absorb visible light, okay, and they have enough energy now to oxidize water to oxygen, okay. Earlier they did not have enough energy to oxidize water to oxygen, okay, because they are absorbing at very longer wavelengths, okay. So this conversion, okay, so shifting to smaller wavelengths, it has nothing, nowhere near green, okay, because to observe green they select a completely different pigment called retinol, okay. And retinol was not selected therefore for plants, okay. Probably they did not see retinol, green light, so they did not take retinol. If they had added retinol along with chlorophylls those days, okay, probably you have black bacteria, because they are absorbing all emission light and also absorbing green. If you see the structure, if you think of organism which have this and absorption here like this, okay, from here to here to here to here to here, okay, all elements of light are absorbed, right? So chlorophyll would have been black, right? But it was not black. Why it was black? It's because they absorb all the elements of light, okay. So and then once chlorophyll is selected, okay, from bacterial chlorophyll, actually, okay, the structure is microcycle has no change, only the peripheral, as I showed you, there is only change in the peripheral side chains, right? I told you that yesterday, right? Only side chains have changed, side groups have changed, no change in the macrocycle of the chlorophylls through the billions of years of evolution, right? So the structure, therefore they could not absorb the green light. They changed, could not. Okay, they came out some other pigments like carotenoids and all that, but carotenoids doesn't make them black. Carotenoid doesn't make them black. They absorb 470 nanometers maximally. Okay, carotenoids. Okay, so that's okay. Okay, and plants got used to for the green light out. Okay, and therefore they you see, even the observed energy we plant has with this mostly blue and red, right? The major two wavelengths where absorption takes place, blue and red. Okay, this blue and red absorption is sufficient for the plant actually. Okay, because it has so much energy now. Okay, it produces NADPH, right? Reductant is producing, okay. Then limitation is Rubisco. Limitation is Rubisco, okay. Always Rubisco is a limitation, okay. Because Rubisco is a sluggish enzyme. The turnover rate is very low, okay, very, very low. Can you give a very fast enzyme turnover rate? When I say Rubisco is very small enzyme, sluggish enzyme, I don't need to have written here. I don't think I have written here in the slide. It is not here. It is not here. Yeah, this is something else about the slide. No. This is slide other one. There are other slides I can show you, but there is no need for me to show that. So there is no need for me to show that. Okay. Carbonic anhydrase is a very fast enzyme, high turnover. What is turnover rate of an enzyme? Yes, binds to substrate, converts the product. Once convert product released from the enzyme active site, they again ready to bind to a substrate, right? Again form the product released from the active site, again bind to the substrate. So how many times it can bind to the substrate forming product. So how many times it can bind the substrate to form the product, okay, per second, determines the turnover rate of the enzyme. Okay, all right. So, okay, so when a sluggish enzyme means turnover is low, means it cannot form the product. It can very fast, Rubisco. Okay, it is very slow. Carbonic anhydrase is another enzyme which is very fast. Carbon dioxide plus what carbonic anhydrase does? CO2 plus water from bicarbonate, right? Bicarbonate. That's the product of carbonic anhydrase, right? And that is very fast reaction, very, very fast reaction. We called it, if we call Rubisco a rabbit, Rubisco is a, what is not rabbit, what is the race was between, not crocodile. Yeah, that mollusk, what is that? Turtle, right? Rubisco is a turtle which moves very slow, right? Carbonic anhydrase is a vehicle moves very fast, is like a rabbit, right? Runs fast. Okay. All right. All right. So this is our, okay. So what I said you, CO2 concentration in atmosphere is increasing, right? CO2 concentration in atmosphere is increasing. And temperature is also increasing. When CO2 concentration is increasing, what is going to happen? What is going to happen to the plant productivity? And plant productivity is going to increase. Why? Because photorespiration is suppressed. Because photorespiration is suppressed. Okay. The Rubisco, there is always a competition between oxygen and carbon dioxide. Okay. All right. So suppressed. But this increase not always. I've written down somewhere here for you. Not always true. There is also down regulation of increase in carbon dioxide concentration, okay, to result in stimulation in photosynthetic carbon assimilation fixation between 30 to 50%. Okay. Primary due to reduction is photorespiration. Okay. Either the Rubisco carboxyl reaction favored in this condition. Oxygenation is suppressed. Okay. But it's not so. However, many plant species, grow elements you do not have, engaged photosynthesis goes up 30 to 50%. Okay. Why do you think so? Why that happens? When you have more carbon dioxide in the atmosphere, okay, and less of oxygenation, more of carboxylation, then plant thinks, why should I invest so much my energy? Okay, one protein called Rubisco. Okay, I can diversify my amino acids to some other useful proteins. Okay, do you understand that? The plant therefore reduces Rubisco content in its own body, in the leaf. So Rubisco concentration very often, not always, but many times, many species are reduced in high CO2 environment. Okay, if the Rubisco concentration decreased, then you will not get a very increase in photosynthesis. Even if oxygenation is suppressed and carboxylation is favored. Okay, in high CO2 environment because rubisco is suppressed. Its substance is less than this figure. This is probably because the plants are able to exhibit acclimated down regulation, decreasing photosynthetic potential, particularly long-term growth related CO2. This anaerobic active response is often correlated with increased carbohydrate levels to get the reduction in total nitrogen rubisco activity. Okay, nitrogen content decreases, protein content decreases. Okay, so it is not only even high CO2. Listen to me. High CO2 in climate change, high CO2. Okay, although we have more carbohydrates synthesized in the plants. Do you understand that? More carbohydrates synthesized. So for, let's say, per grain, we had 1 grain, let's say, weighs 20 grams or 100 grams. 1 grain, let's say, weighs 10 grams. Okay, 10 grams of one grain, let's say, has 1 gram protein. So 10% is the protein. Okay, all right, maximum in a cereal. Okay, all right. Now, when plant assimilates more of carbon dioxide, okay, manufacture more and more carbohydrates, right? More and more carbohydrates. So the what is nitrogen? Where comes proteins come nitrogen? Where nitrogen comes from the soil in form of nitrate, right? So nitrate assimilation, nitrate reduction to ammonium, and subsequent assimilation of ammonium to amino acids. Okay, okay, cannot keep pace with the increased CO2 concentration fixation. Increased CO2 fixation. CO2 fixation is high, the carboxylation is high. Okay, but so we have more carbohydrate, but you cannot synthesize amino acids and amino acid proteins at faster rate because of limitation nitrogen assimilation. Okay, nitrogen content in soil not increasing. What is increasing? The CO2 atmosphere. You may have some, but if you increase the nitrate input into the soil, okay, increase the fertilization level in the soil, probably you will not have a reduced protein content. Okay, but in natural ecosystem, okay, natural ecosystem, the tree or a plant or any other plant, okay, you have carbohydrate, you have in a seed you have more carbohydrate but less protein. You understand that? Huh? Because amino acid content, nitrogen assimilation will be low. Okay, nitrogen assimilation means same, it is not going to be reduced. Nitrogen assimilation will be same. So protein synthesis will be same, but there will be more carbohydrates because of CO2 concentration atmosphere is increasing. So you have more carbohydrates and less protein in the seed. So wheat plant growing in nature will have more carbohydrates and less protein. They are not the carbohydrate seeds in elevated CO2. So this may lead to protein deficiency in the plants. In the seeds, of course, we may face a protein deficiency later on. Okay, but all these things are good enough if it's not accompanied by high temperature. But as I told you before, high CO, and you know by now, high CO2 is going to accompany by high temperature. Okay, then what is going to happen? Tell me, what is going to happen? When high CO2 and high temperature, what is going to happen? Think and tell. Huh? Huh? Enzyme? Enzyme? We are not going to denature in phase. Enzyme denaturation is very in high, very high temperature. Okay. Huh? that is viscoactivity and all that will be affected high temperature. But that is a different story altogether. Reproductive growth will the vegetative growth will higher. Okay, you grow at high temperature. Little bit high temp, 1 or 2 degrees centigrade increase. We're not going to have 10 degree increase in temperature. Okay, we will have a 2 to 3 degree, maximum 3 degree increase in temperature. That will may favor crop growth. Okay, but it will reduce the reproductive growth. Pollen grain setting will be reduced. Pollen grain germination will be reduced. Pollen grain formation will be reduced. Okay, when pollen grain formation is reduced, what is going to happen? Fertilization will be less. Okay, flowering fertilization less, so less seed formation. So you will reach a scenario where you have more vegetative growth and less reproductive growth. So what is going to happen? You have more vegetative mass, but less seed formation. So it is going to impact our agriculture if high CO2 is accompanied by increased high temperature. You see, 2-3 years ago, in month of February, the temperature went to 30 degrees in northern India, northern belt, but a lot of wheats are grown there. Wheat is grown there in northern areas, in Punjab, Haryana, all that places, Uttar Pradesh, and all that. Okay, they grow wheat, and wheat flowering time is in February, January, February. February temperature increased by 4 to 5 degrees centigrade. Okay, average temperature of February increased 3 years ago, 20 to 5 degrees centigrade. So that led to reduction in pollen grain formation, reproductive decrease, and reduce seed production of wheat. Okay, and therefore seed production was less. Okay, because pollen grain didn't germinate, pollen grain was not formed or did not germinate on the stigma. Okay, all right, no fertilization, therefore no seed formation. Okay, so it's highly reduced, highly suppressed. So even 2 to 3 degree increase in temperature may not create a big problem for vegetative growth of the crop, but may affect the seed setting, flowering, and pollen formation. So we have more biomass and less seeds. Okay, so the advantages of high CO2 for favoring more vegetative growth will be gone at high temperature, especially for wheat, even for rice. Same thing, 3 degrees centigrade is more than enough, average increase 3 degrees centigrade. It also will decrease the reproductive growth in rice also. Okay, and this is going to happen. Okay, because we've already seen increase in temperature. Okay, right? temperature बढ़ा है कि नहीं है? बढ़ा है ना? How much? कब बढ़ा है? कितना बढ़ा है? I don't know, I have that slide with me. Here it is. Here you see Arctic region. What is Arctic? Northern pole. North pole is Arctic, right? The temperature measurement in Arctic. This is measured by NASA, 1880, and I have figured it 2004, but it is 25, is much higher. You can see the temperature constantly increasing here, right? This is minus. If this is today's 0, the minus 1.5. Okay, we are giving that to crossing 0, going up and up and up here. I'm sure you've gone up by now. Okay, so Arctic has already experienced around 3 degree increase temperature. Arctic region, not of the whole world, but the North Pole. North Pole has already increased its temperature last 120 years. Okay, 1 to 3 degree increase temperature. Okay, therefore, glaciers are melting, and all that. Why do you think Arctic is increasing more than Antarctica? Antarctica temperature is less increase. Arctic is more, North Pole. More of human activity there, more human activity in Baltic, Northern Pole, North Poles, than South Poles? No? What? Hmm? Yeah, ozone layer depletion is there, but that may not affect much of temperature. It will? Huh? It will? Why? Why? Huh? UV radiation is more. Of course, UV radiation is more there. Okay. Huh? Arctic black. Arctic, there are a phenomenon called albedo. Albedo. What is albedo? They are all ice, right? All ice, right? And when light is falling, also going up immediately. Okay, okay. Light is striking, and in the ice, reflecting back. Okay. So if it reflect back to the atmosphere, it's fine. Okay, but it's observed on Earth, then temperature is going to increase the temperature. Okay, it goes back fine. Okay, so this if you have green leaves there, plants there, it will have positive effect there. Okay, you observe light and increase the temperature. Leaves there, we're going to observe the light and increase the temperature. You understand that? So we don't want in North Pole region, polar region, where albedo is working for us. Okay, it's coming and going. Okay, not being observed by Earth. Okay, okay, getting reflected back to atmosphere. Okay, if that is changing, okay, okay, it is reduced. Reflection is reduced by absorption of green plants. Even small amount of green algae that is growing in the Arctic ice would change the albedo, change the reflectance. That will increase the temperature. Okay, that is one of the impact. Okay. Okay. So, do you understand what I am telling about this aspect? I don't know. The recruitment of these three proteins, you know about this? Oxygen evolving complex, right? You know oxygen evolving complex. What it does? I think he must have taught you by now, your Japanese collaborator. Did you talk about anything about that? Oxygen evolution or oxygen uptake? No. Okay. Takahashi, na? Okay. What he taught you? LSC. Huh? Structural biology. Oh, yeah. This is also part of structural biology. We know about manganese for complex structure and all. Okay. I'm sure we'll talk about it, so I'm not going to talk about it. So I have left it for him. Okay. Now, another point I must say, yes. So this is how to combat elevated CO2. Okay. Reduction of greenhouse gas emission, of course, very important. CO2 emission in particular, and then methane, right? Methane emission. Methane emitting today very minimal, very low, but still some amount of methane is emitted from the rice fields. Why? Why? Because water, rice needs a lot of water. Rice grows in green rainy season, okay, and the earth is full of water, waterlogging. So there is no oxygen on earth. The air occupied, the space occupied by air is now filled by water. Okay, so there is no air. So there, what will grow? Which bacteria grow there? Anaerobic bacteria, right? So anaerobic bacteria will grow where? In the rice field. Okay, in anaerobic conditions. So they will emit methane. Okay, right? So the methane emission also causes global warming. Okay. So you have reduced the greenhouse gas emission. Increase CO2 for plant, especially reforestation. You have cut down so much, 70% of the forests. Okay, cover. Can you restore it back? It's a very important problem. As I told you, you restore it, we will certainly decrease temperature. Certainly. There is no doubt about it. Okay. You plant trees, enormous number of trees, all around where you're staying, around the globe, wherever you can. Okay. Will certainly reduce the temperature. Certainly. I told you the reason why, right? Because I told you the plants transpire and latent heat evaporation is provided by the leaves, and therefore it cools the atmosphere. Okay. Plantation of fast-growing trees is very important, like Populus deltoides or some other, or many other species. But today, Populus deltoides may not be a great choice, but any tree is fine, may not be fast-growing, but they should be strong enough. Because, you may experience this, for coastal India, experience always what? The many hurricanes, right? Right, hurricanes. And these come and devastate their trees. Okay. And that is mostly due to the fact that there are, many coastal plants, mangroves. Okay. Mangroves were there everywhere throughout the, if you cut from Bengal to Cape Comorin, Kanyakumari, back to Gujarat coast. Throughout. Okay. We had mangroves. Okay. You have destroyed mangroves like anything. Very few pockets in India have now mangroves. One probably in Sundarban, another maybe Bhitarkanika in Odisha, and another maybe Pichavaram in Andhra Pradesh. Huh? Kerala also have some mangroves. Some mangroves. That's it. Goa may not have it also. There may be some mangroves in Goa also, but not much. Okay. So if three few localities, not mangroves in Kerala, mangroves not everywhere. In some places. That's what I'm saying. Not everywhere. This is the mangrove forest. Do you see this? of Sundarban. Okay, anybody have gone to Sundarban? These dense forests you will not see anywhere else. Vitakhari mangroves are there, but not that dense. It's there, not that dense. Okay, Sundarban is full of mangroves. Okay, Vitakhari mangrove, but a small place. And Sundarban is very big, is a big, vast place. Okay. So, what we are doing, look at the roots. Look at their roots, how they are holding the soil. And when air comes, hurricane comes, they remain as wind barriers. Here, Sundarban also a lot of mangrove destroyed. Okay, they used a huge wind barrier, 4-5 kilometers long width, wind barrier. Okay, so they could reduce the intensity of hurricanes. Okay, intensity of air, speed of air. Okay, and when air reaches the landmass, okay, its speed was reduced. Therefore, it was not impacted much. Okay, and what is happening now? They are all gone. So we are all subjected. All coastal belt is subjected to very high speed of wind. Okay, and that is destroying, uprooting all the plants, all the trees. Most of the trees are gone. Okay, the big trees are gone in hurricanes. Okay, so that is one of the reasons why there is also global warming. When coastal belt is so cool, now it's very warm. You people are from Utkal University, right? So it is very warm there, very hot there now. You're not there when we are studying there. Even we are studying there in Bhubaneswar in 70s. It will be hot for a while, for 2-3 hours during daytime. Then evening, it all, even afternoon at 4 p.m., it will so windy and all that. Cool, hot is gone. And nights are very good, pleasant, evenings are very pleasant, no problem. Only 2 to 3 hours, it's from 10 to maybe 4, 11 to 4, 4 to 5 hours. Then it's gone. But now all the time I have hot air in Bhubaneswar. That is because it is full of concrete, number one. Got all the forest gone from Bhubaneswar. All the forest, and the 99 cyclone came. That was destroyed all the forest, big trees, okay, from everywhere. 99 cyclone, and followed by many cyclones, okay. And they have all destroyed the forest cover, and whatever forest remained, man-made invasion of cutting down the forest and building buildings back into concrete and change the temperature. Okay. See, Earth has a big light absorbing power. Okay. But concrete doesn't have that much power. Okay. The infrared light, it absorbs very deeply and it produces heat. Infrared light, it produces heat, na? This is another component, so reflection in the urbans. These are respiratory roots. Do you see the respiratory roots? Huh? Nematophores. Yeah, coming from the soil. They are respiring in salt habitat through the nematophores. They are roots. Roots need oxygen, na? So roots are respiring through the nematophores. Okay, because they don't have oxygen in the soil, all occupied by water. You see the root system, conserving soil, trying to conserve soil. Okay. can we come to an alternate fuel like hydrogen, right? When it's a green fuel, alcohol is not a green fuel. We are trying to mix alcohol with petrol, right? Petrol with alcohol, right? To reduce the consumption of petrol, doesn't matter. But still emit alcohol also burn to carbon dioxide. Okay, so it's not a really green fuel. It's also producing carbon dioxide. We're painting green, I don't know why. Okay, all right. One reason being that it is coming, but here the CO2 is recycled. CO2 is absorbed by plants, right? You produce alcohol from the plants, then alcohol is consumed again. So same carbon dioxide is sent back to atmosphere. Okay, so it doesn't lead to reduction in CO2 concentration from the atmosphere. So and in a country like India, how much grain we can destroy? To make 20% alcohol, blend 20% alcohol with petrol. Okay, probably we have to destroy 2 states like Kerala. Okay, 2 states area-wise. Okay, grow all crop. Okay, and that crop you are producing alcohol from there. Okay, area-wise. Okay, then only you can probably meet the demand of alcohol. Okay, to be blended with what? Petrol. So that is not a feasible program, sustainable program. Okay, not only that, you are destroying the land mass for what? You are using nitrogen from the soil, all the nutrients from the soil, and doing use to burn it as in the fuel to carbon dioxide. So the amount of nutrients you need for your survival as food is being depleted from the soil. Isn't it? So you are having more and more fertilizers, and they're creating more and more harm to the soil. Okay. So instead of doing that, if you think of hydrogen as an alternative fuel in car, hydrogen fuel. Electric vehicles are okay, but electric energy is not coming of your own. Electric energy also coming either from thermal energy, burning coal, okay, or hydroelectricity, making vast devastation to the landmass, right? Making many people displaced, okay, when you are making water reservoirs, okay, all right. So it is, and also in Himalayas, it's creating a lot of havoc there now. All landslides are taking place in Himalayas, all country landslides, huge landslides, because you make dams, and water it accumulates there, and the water percolates into the rocks in the mountains, and then rocks become slowly comes out. Okay. Okay, the adhesion of the rock between rock is gone. So then what happens? The rocks fall down, land mass fall, big landslides take place. Okay. So it happens. Okay. All right. So if let's say like this. Hello. Good afternoon. I am not there till tomorrow. How do you produce hydrogen biologically? Can you produce biological photosynthetically? at the expense of solar energy. Hydrogen can produce photolysis water. Photolysis water, but you don't know, put electricity, pass electricity in water, will convert hydrogen and oxygen. You can get hydrogen from there. A vast ocean, no problem. But energy input is quite high to hydrolyze water to oxygen, water, hydrogen, oxygen. So idea is, can you do it at the expense of solar energy? We are receiving, okay, directly from the what? Directly from water. With photolysis of water to photosynthesis, right? So the enzyme called hydrogenase in many algae. Chlamydomonas is one of them. Very important, okay? One of them. What it does, once ferredoxin is reduced ferredoxin, okay, then hydrogenase, okay, converts reduced ferredoxin to what? Oxide ferredoxin hydrogen. Here reduced ferredoxin does not give electron to NADP, for NADPH. Rather, it gives reduced ferredoxin gives electron to is now oxidized to hydrogen. Okay. Alright, by enzyme hydrogenase. Reduced ferredoxin, you know, from photosynthetic electron transport chain. The ferredoxin by FNR, ferredoxin NADP reductase, gives electron to NADP to make NADPH. But before it gives electrons to NADP, you give what? Hydrogenase, which is already present in the plant, in the algae. Okay, so it gives hydrogen to it. Okay, okay, it's so simple to say. So this is what is it, right? Let's say pH2, right? Water for 4 protons, right? Now, this what happens under normal conditions, right? NADPH production and all. NADPH conversion. If you make it a little bit anaerobic atmosphere, okay, little anaerobic atmosphere, then what will happen? Let's see. Reduce ferredoxin here. Reduce ferredoxin, right? Ferredoxin works as hydrogenase to form hydrogen on anaerobic condition. Okay? Because hydrogenase survives only anaerobically. Hydrogenase enzyme is destroyed by oxygen. Remember that. Hydrogen is destroyed by oxygen. So if you can maintain a little bit anaerobic condition, okay, in the cell, okay, then you can produce hydrogen. Okay. So we are talking about this production of hydrogen, anaerobic conditions. How can do that? Yeah. So there are fuels, oxygen microbe, hydrogen fuel cells.
Excuse me, sir. Sir, excuse me, sir.
Yes.
Just wanted to sir, in case of this oxygenic photosynthetic hydrogenases, like enzymes, hydrogen producing enzymes, they are sensitive to oxygen. So, how about this anaerobic photosynthetic bacteria, which are usually grown in anaerobic photosystems?
Anaerobic photosynthesis, okay, if you can, that is a possibility.
Yeah.
But let's say you give hydrogen sulfide as the electron donor, H2S,
yes, yes,
or something like that. It depends on what kind of bacteria you are selecting.
Yes, yes, yes.
Then it can produce hydrogen, no problem in that. Anaerobically, because they are anaerobic bacteria, photosynthetic bacteria. what the issue is that you are again giving substrate like hydrogen sulfide. It is not water, it is substrate anymore. It is hydrogen. You are manufacturing, producing. You have to give some. You to synthesize hydrogen sulfide. You have to spend some energy. Okay. It is not all prevalent in water.
Yes, sir.
Or everywhere in ocean. Okay. Water is the very cheap source of electron donors. But whether acetic acid, acetate, hydrogen sulfide, or any other substrate, then you have to provide energy for it. So energy input is very high than your output. So water, vapor system, reduction, right? This is hydrogen, right? Okay, hydrogen production. Okay, and you can do that. How can you maintain climate luminous in a hydrogen low anaerobically? So what is mitochondria does? Respiration, right? It consumes oxygen, right? Making water, anyway. So what you do, grow them in sulfur starvation. Sulfur starvation. Okay. Plant, you are reduce the sulfur from the medium, from alga. We are growing alga, but don't give sulfur to it. Okay. Or very, very minimal sulfur. Okay. So under sulfur deprivation, you can see in hours, you can see the rate of photosynthesis will drop, okay, because there is no sulfur. The rate of photosynthesis is reduced, okay. Whereas respiration is not reduced drastically, okay. So at this point, here, the anaerobic environment or not. Respiration is high, photosynthesis is low. Under sulfur deficient condition, you can see photosynthesis drops. Okay, respiration remains almost the same. Okay, so at this point, after a few hours of growth or sulfur deprivation, you will see rate of respiration will go high, rate of photosynthesis will be low. So this from time period from year to year, from 40 hours to 120 hours. This is the stage where there will be anaerobic growth, anaerobic medium. Cell will be anaerobic condition. Isn't it? Photosynthesis is high, respiration is photosynthesis low, respiration is high. Right? Understandable or not? Right? Oxygen production is less, oxygen consumption is more. Oxygen consumption to respiration is more, right? Photosynthesis production, oxygen through oxygen production is less. This is all anaerobic environment. So in anaerobic condition, as I told you, hydrogen can survive. Hydrogen is functions. Hydrogen function under anaerobic environment. Okay. So there is hydrogen production. So if you measure the gas volume per ml and you measure under sulfur deprivation, you can see the hydrogen gas produced on the AA top from 50 to 100 hours. Amount of hydrogen produced. And this is actually you did in your lab, and we collected the carbon cell. You grew it at sulfur deprivation conditions, and you see, and we collected hydrogen by downward displacement of water. Okay, hydrogen collected from in the top. You see that? Huh? Hydrogen collected from by downward displacement of water. Okay, so you can do it, but economic feasibility of it, that is the point. Okay, you grow under sulfur deprivation, okay, algae, okay, and put the and collect gas from here, and you see in the top there is a hydrogen gas by downward displacement of water. Okay. So there are many ways. So if now you collect hydrogen and put it into your car, okay, what will happen? Hydrogen will be born to consume, hydrogen oxidized by oxygen to water. So you get hydrogen, so what the byproduct is water. Hydrogen peroxide will become water. No carbon dioxide. Right? Hydrogen is the fuel driving your car. Okay. That possibility is there. It can be done, achievable target. But you need a lot of input for it. Okay. This is one way you can do it. There are many other ways you can also do it. Okay. But the hydrogen is the problem, the hydrogen is sensitive to oxygen. If you can genetically engineer hydrogenase, okay, which is insensitive to oxygen, okay, then no problem. So you knock out the hydrogen from Camda monas and put genetically engineered hydrogenase back into the same system by transformation again on any promoter, 30-base promoter, whatever. Then it will be, you have now hydrogen, the algae does not have any original hydrogenase, that is gone, the gene is gone, and supplemented with what? A hydrogen resistant hydrogen, oxygen resistant hydrogenase. You see, this is not hydrogen, not only enzyme. Can you see any other enzyme which is resistant, which is sensitive to oxygen? Any other enzyme? Well, nitrogen is one of them, yes. Nitrogen is also needs, is sensitive to oxygen. So, if you see nitrogen production, nitrogen fixation, what they do? Nitrogen, what they do? Convert nitrogen to ammonium, right? Ammonia, right? Convert nitrogen to ammonia, nitrogenase. And this nitrogenase is inactivated by oxygen. Therefore, you see, if you see cyanobacteria, they have heterocysts, right? Heterocyst deficient photosystem too, so there is no oxygen evolution, okay? No photosynthesis, no oxygen evolution, and a very thick wall, so that the atmospheric oxygen doesn't enter there, and whatever oxygen there is consumed by its own respiratory system. So you become a real anaerobic environment, heterocyst, where nitrogen is converted to ammonia, or enzyme nitrogenase. Read it. So in nature such system exists. So leg hemoglobin also, leg hemoglobin also fixes some nitrogen in the where? The nodules, root nodules, mediated by rhizobacteria. Not it. They have also, but they create an anaerobic environment there to fix nitrogen to ammonia. So you need, for hydrogen function, nitrogen function, you need anaerobicity. Okay. If you create a condition of anaerobicity, okay, or you can genetically engineer hydrogen, okay, where it is insensitive to oxygen. It's extremely difficult to do it, but it can be done, I guess. Okay. Then no problem. You can produce hydrogen from water without fear. Some of them will get consumed to, it may happen that some of them will go to produce hydrogen, some of the electron protons may go to fixed NADP, NADPH, carbon CO2, CO2 fixes. You never know. Okay. All right, to stop here. Okay.